I'm a different, in a good way, speaker for the Friday seminar. This is one of our earliest graduates of this program. Anamaria has 2 PhDs. She came in with a PhD in economics. And then we fixed her. So now she can do computational social science, interdisciplinary, and has bounced around in several different places doing computational social science related stuff. And she's going to tell stories. I'm not going to go embarrass her anymore.
Thank you, Bill. Thank you for being here, especially on a Friday afternoon. So as Bill was saying, I'm Anamaria. I graduated from here. It's good to be back. It's good to see some familiar faces. What I would like to talk to you about today is a project that I participated in last summer. And this is also a little bit of an unusual research project because unlike the usual projects where we have hypotheses and we test them, this project is much more exploratory. So basically, I would like to take you today on a journey to find some answers to one of the most important questions in science. Basically, where does life come from, and is life in the universe ubiquitous or rare? So, as any traveler that goes on to explore uncharted territories, which is the case for this project here, I am going to show you some of the tools that we used and basically how we relied on the wisdom and the knowledge of some of our predecessors in science in terms of physics, astrophysics, geochemistry, chemistry, biology, astrobiology, data science and computer science, we are basically going to take this journey in an interdisciplinary fashion. So unlike other research problems, as I was mentioning, where we formulate hypotheses, we build models, and we validate our assumptions based on some evidence, these big questions, such as is there life elsewhere in the universe, start with an exploration and with an element of discovery, because we don't know exactly where we are going, and we don't know what we might be finding along the way. Therefore, in many respects, these kind of questions need an even more robust scientific scrutiny than usual in terms of methods and theories that we employ. You're probably familiar with the name Carl Sagan, and as Carl Sagan used to say, extraordinary claims require extraordinary evidence. Therefore, where do we start in this journey, and where is our North Star? And we are starting with this research accelerator. NASA, in partnership with the SETI Institute, have this program over 8 weeks over each summer at NASA Ames. It's close to San Francisco, called Frontier Development Lab. At the NASA Frontier Development Lab, data scientists and planetary scientists come together to solve in a very rapid fashion, Silicon style, some of these big major problems. Basically, the NASA FDL teams are formed of 2 data scientists and 2 planetary scientists, and they have mentors. And for a period of time of, like I was saying, 8 weeks, so it's even less than 2 full months over the summer, they need to reconceptualize some major problems, such as can we forecast the next solar flares? Do we know when the next asteroid is going to hit Earth? Do we know what the shape of a particular comet is? And so on. And thank you. And so we start by even reconceptualizing these problems and taking them all the way to a good place, a place good enough where it can be implemented by scientists. And this is the team that last year worked on this astrobiology problem. So this was one of the 8 teams. Aaron, Will, Adi, and Rod and Will Rod and Aaron having been the data scientists, and Will and Adi Will being a physicist and Adi being an astrobiologist. So together with them and with some other mentors that I mentioned along the way, we started tackling this huge problem: what is universally possible for life? And I want to ask you if you know what kinds of data scientists are out there, if you know. So there are two kinds of data scientists: those that can extrapolate from incomplete data, and those that we don't know, right? And this particular joke is very relevant here, because for this particular project in astrobiology, we had a lot of incomplete data, and we had a lot of extrapolation that we needed to do. As I was mentioning, NASA Frontier Development Lab is an AI research accelerator. So if we are framing this question in terms of an AI accelerator for astrobiology, and we are emphasizing the word AI, which is broadly construed as using mostly deep learning, neural nets type of algorithms, we are actually facing a problem. Because, particularly in astrobiology, we have a sample of one when it comes to life. We don't know about life elsewhere in the universe. So where are we going to get all these data that we need for AI algorithms? Okay. So the primary question is not, well, for us, was not exactly which deep learning model to apply, what kind of neural net, but how do we get more data? How from one, the sample of one, we can have a sample of many. Astrobiology is the study of life here on Earth and on exoplanets, but also it can be the study of life at different phases of the Earth in the geohistory of the Earth. And in the first week of the program, we started basically by, again, reconceptualizing the problem, so charting the seas. And we thought about how can we tease out, what would be most fruitful, most useful to make headway with this problem. Can we simulate and explore non-biological nucleotides? Can we test for shadow biospheres? Shadow biospheres being those biospheres that we don't, where life is not DNA based. Can we simulate using some? Can we do some simulations using the LIGO principle? And so on. Can we find the extreme limits of life? Are there extremophiles? Even imagery, if we don't have exact data from the biology, can we just use imagery, and can we identify fractals in imagery? Will that be helpful? So for actually 1 week or even more, we just started to reconceptualize this problem. And basically the methodology that we ended up using is, okay, we reduced the search space into, can we take Earth as one instance and example of life and split it into several observations, but dividing the lifetime of Earth into its epochs? Can we establish strict objective universal principles about life itself? How can we explore the complexity of how biology and the environment change and co-adapt and co-evolve with each other? And can we extrapolate from the habitat level of biology to the planetary and atmospheric scales? And how do the diverse samples of life here on Earth from different biospheres, what can they tell us about the boundaries for universal life? If we are looking at extreme temperatures, at extreme life energies, and in general at extremophiles. And we ended up going back to what is called the NASA Astrobiology Roadmap, which is a roadmap for that NASA puts out periodically every few years about how we can proceed about searching for a life on other planets. And particularly from all these points that the roadmap has, we thought that our project, that we can make headway by looking, one, at the coevolution of life and the physical environment, and here is where complex systems and complexity science come in, and constructing habitable worlds, and this is where data science and simulations come in. And again, considering the NASA Astrobiology roadmap, we thought we would be able to actually give some answers to goals number 1, number 6, and number 7 here, understanding the nature and distribution of habitable environments in the universe, understanding the principles that will shape the future of life, both on Earth and beyond, and determine how to recognize signatures of life, named biosignatures, on other worlds and on early Earth. So here, basically, we have an opportunity to link biology and geology, or biology and environment, into an interaction together that apparently has not been done before. It has not been done at NASA, and it has not been done elsewhere before. So we basically made headway by clinging to the idea of the coevolution between biology and chemistry, particularly geochemistry, and looking at how these interconnect with each other from micro to macro. So this is officially the framework for planet habitability. What does this diagram here mean? It means that there are lots of coevolutionary and codependent processes that contribute to a planet's habitability, from astronomical, physical, astrophysical processes, but so far biology has been missing in this planetary scale modeling, right? So until now, the scientists have looked at planetary habitability based on how far away is the planet from the star, what kind of star is in that particular solar system, what kind of geology or what kind of chemical composition the planet has, but so far no biology. So this was an opportunity for us to actually link these and look at, so explore this nice niche with respect to understanding the link between the microprocesses on the surface of an exoplanet and the atmospheric processes or the macroprocesses of the planet. So basically looking at microprocesses that can be biological, and then looking at the atmospheres of the planets. And we can do that looking at the fluxes from some generalized models of biological processes that are have already been published. And as we started this project looking for data, we said, okay, this is the kind of data that we can use. We can use instrument data from Kepler and more recently from the TESS observatories that are not on Earth, they are up in orbit. Then we can look at the transit discovery. So we can take we can look at the what we know about exoplanets, and so far there have been more than 3,000 exoplanets detected in the universe. We can look at the atmospheric spectrograph. This is a little bit more tricky. This has actually been started to, we can do this only maybe for the past couple of years, so this is very recent. Until now, we could only see whether an exoplanet might or might not have an atmosphere, but we could not detect what kind of chemicals or gases are in that particular atmosphere, right? And in order to infer the biology on the surface of the planet, we can look at, we can do some simulations. Okay? So basically our project, we reduced our project to just looking at the, if we can look at the atmospheres of the exoplanets and whether we can simulate some data with respect to the biological process on the surface, can we make headway about how we can detect biosignatures in the exoplanets data? Okay. So how do atmospheres of the living, co-evolving, and abiotic planets differ from each other? We can look at some distributions. So we can look at distributions between what is biotic and abiotic when, on a planet, on an exoplanet. So biotic processes. Yeah. So biotic processes are those that can harbor life. Biotic processes usually involve having greenhouse gases, such as methane, oxygen, hydrogen, water, and so methane, oxygen, hydrogen, water, methane, and carbon dioxide. Thank you. Yeah. While abiotic processes involve processes that don't they're less likely to harbor life. on any planet you can find a combination of both, right? So you might have might have greenhouse gases plus other gases, but it doesn't necessarily mean that you find life on that particular planet. But the first step would be in order to reduce the search space even more, is to look at those where we can find these biotic processes. And we can do that by looking at the temperature and the frequency of the exoplanets that fall under a particular temperature on the surface, right? So a planet can be very, very cold, where you have, let's say, methane is in liquid state, and you can't really, maybe you cannot have life there, although now with the new mission towards Titan, that might change the equation there as well. But again, a planet can be either too cold, either too hot, so there is a sweet spot in temperature for a planet where you're more likely to find this combination of biotic and abiotic processes, and particularly to look at the biotic processes on the planet.
Sorry, just a quick question about that.
Yeah.
So, but presumably your biotic temperature distribution there would only be a sample of one, because you're at the planet level and there's only one planet that has biotic processes.
That's true. That is a possibility. But again, with so TESS was launched last year, and only now they are gathering lots of data with respect to how many exoplanets are there. And next year, or in a couple of years, they will launch the James Webb Telescope, which will particularly look at the atmospheres of the exoplanets. So, now there are more than 3,000 exoplanets that we know of from the Kepler data. As we enrich and this has been almost like Moore's law in terms of discovery from the 90s, so when you would find an exoplanet, once maybe every couple of years. Now you find many during a year. So given how the data can be enriched with the new telescopes, it's less likely, that you'll find a particular planet in just one niche.
Well, I think the question is, can you subdivide the Earth's epochs and identify extremes where, life is here, right? And it had to be here, I guess, not from the beginning of the planet Earth 3 billion years ago, but 2 billion years, right?
Right.
So there's lots of different environments. That's a lot of different planets.
So that's another hypothesis, whether this life as we know it on Earth is the only life that Earth ever had, or maybe during the late bombardment, which was I think like 2 billion years ago, whether there was another phase of life on Earth. We don't know that. Also, it's possible that Mars at some point was in the habitable zone, and also Venus was at some point in the habitable zone, and then they were pulled apart from due to Jupiter's gravitation. And so I'm not an astronomer or a physicist or a chemist, but working with these guys has been incredibly exciting, and I've learned a lot. So I cannot have an expert level answer to this, but given the statistics and the rate of discovery, and given the constraints that astrobiologists know with respect to what is habitable or not, or what can potentially be habitable or not, I don't think we'll ever have a sample of one. And if we do have just one sample of one, then that is going to be under a lot of scrutiny. Right? I don't know if you know, but in the news, there was a week or two ago, there was in the news this discovery about an exoplanet that has water on its atmosphere. And everybody was jumping up and down. Okay, we finally found an exoplanet that has water. Can that be the second Earth? But actually, as I'll show in a minute, it doesn't matter if you have just water. There are so many conditions that have to be fulfilled with respect to the composition, the pressure, with respect to how far away you are from the star, what kind of star is the planet in, and so on. That you can say yes, it's water, but you cannot say yes, there is life. Okay, so yes, basically we can look at the if we are looking at the atmospheres and at the compositions of the atmospheres, can look at what atmospheres are potentially habitable and what biology could they support. And this is our solar system from Mercury all the way to Neptune. Yeah. And with different atmospheric compositions, and we know that only Earth so far has life. Although we don't know whether the moons of Jupiter and Neptune, whether those have life, right? So that's the next phase of exploration. And what other kinds of data can we generate and look at? As Ken was mentioning, so what constitutes, so where in the phase or in the history of the planet, there have to be at some point concurrent conditions with respect to the planetary environment and the habitability conditions, right? But that can be they can be sustainable only for a short period of time, and then you'd have life disappearing, right? Which may be, or maybe it was the case of Venus, for instance, and that's called a Gaia bottleneck. So in order for life to survive and to thrive, you have to have these conditions between the planetary environment and the actual habitability conditions to co-evolve and coexist with each other for a longer period of time. So yeah, so one is Gaia bottleneck, the other one is called Gaia regulation. And on Earth we can say that we have Gaia regulation. We don't know about other planets where we might have had these Gaia bottlenecks. So with this data we can therefore explore both the past and the future conditions of life, so not just life on the current exoplanets that we know now. In our particular project, so we started simulating the data, right? And we ended up using 3 different models to generate 3 types of data and linking them together. The KEGG data, K-E-G-G, is a database of chemical compositions that are being used for metabolic fluxes. Okay, so this is the chemistry of metabolic fluxes. So we use that, and we use that into a model of metabolic fluxes of E. coli, which is the most studied organism on Earth for different reasons in biology. Then we used an agent-based model. We didn't design it, but we adapted it and we used it. It's called ExoGaia. It was developed by Tim Lenton. An agent-based model that simulates the biomass of a planet based on the planet temperature and albedo. Albedo is the refraction of the light of the planet from the star. And the third one, we use the Atmos NASA simulator of atmospheres that basically simulates planetary temperatures and pressures based on various combinations of gases. So this is how we link basically from the most micro processes to all the way to the macro atmospheres of the planets. And then we infer back, if we detect the atmosphere of a planet, can we infer all the way back what kind of biological or chemical processes happen on the surface of the planet.
And you?
Let's see. All right. So, step 1. We selected the 6 greenhouse gases that we know of, and scientists say that this is how you're more likely to find life on other planets, to create the stable and warm atmospheres that we need, and these would be methane, oxygen, carbon dioxide, hydrogen, water, and nitrogen. And we use this molecule composition as an input into the Atmos code, and as an output we had the full atmospheric profile for each of these atmospheres. So step 1, generate the atmospheres. The first step actually into looking at the atmosphere ingredients and simulating the temperature, the pressure, the mixing of ratios of gases, the gas fluxes required for consistencies from Atmos was actually to translate code. Because NASA works in Fortran, besides spending a huge time or conceptualizing the problem, we also had to spend a lot of time into actually translating code from Fortran to Python. We actually had the help of Google for this, not for actual translation, but they gave us all the resources that we need basically for using Kubernetes, for using for dockerizing, and for doing all these simulations in the cloud. So Atmos is basically this one-dimensional photochemical climate model where you can simulate the photochemistry and the climate of terrestrial planet environments. Okay? And we coupled the code. I'm not going to go too much into details but what we ended up doing is basically looking at different mixes and ratios of these compositions. We ended up looking at what is the surface temperature on a planet. And for instance here, based on different compositions of methane, we could see what was the temperature of the planet, how it was increasing incrementally. And then we of course looked at combinations of gases. So in this particular simulation, we looked at combinations between carbon dioxide, methane, and water. And depending on different combinations of these three gases, what type of temperature we can expect to have on the planet, but also what type of pressure, right? Because temperature and pressure at the climate level, they go hand in hand, right? You can have the right pressure, but if you have the right temperature, but if you have the pressure too high or too low, then you don't have the exact chemical combinations that you are looking for. Right? So here is basically the distribution of the potential exoplanets based on these various combinations of gases and planetary temperatures. And for example, given a water profile of the atmosphere from 0 to 1, most exoplanets would exhibit very high temperatures, right? About 40 degrees Celsius. And to give you some reference, Earth is around 15 degrees Celsius and has a profile of about 1% water and about 0.04% carbon dioxide. So that was the first phase of the project. So we simulated that. Then the second step of the project was to look at the micro processes and to simulate these metabolic networks. As I was mentioning, KEG is a database of biochemical reactions that shows which chemical molecules are reactive to which ones and what kind of quantity of biomass they release. So essentially it's a matrix, right? So you can create a network. So it's a matrix of approximately 6,500 biomasses and 5,400 reactants. And what we ended up doing is looking at that and then also randomly generating genomes. So not real genomes, but synthetic genomes that modulate any combinations of the 6 greenhouse gases and check the output of what would these gases would be. So basically, okay, if we have these synthetic non-existent organisms that we know that they use these chemicals, what kind of chemicals do they feed on, do they release, so that we can understand the relationship between the atmosphere and these potentially existing, but now existing only in the computer kind of genomes. So we looked at the metabolic networks of, metabolic fluxes of these genomes that we created. We created about 10 of them. So we created a genome that could feed on all the gases, right, all the greenhouse gases. We created genomes that could feed on each of the gases or on different combinations, and we also used a baseline. Right? So in this particular case here, we can see that what we call the universal E. coli, which doesn't exist. But we think of it as this organism that can feed on all the greenhouse gases. So it has a wide range of adaptability, right? It has more, it has an enhanced metabolic flux than the real E. coli that we can find here on Earth. And then we also looked at the, as I was mentioning in the chemistry of these metabolic networks, that you can create networks, right? So we can look at the network of how different chemicals can react with each other in the real E. coli and versus the universal E. coli. And they are surprisingly similar, right? So we looked at the network density, how much what is what is the connectivity, right? So how many links can you find between these different reactants in real E. coli versus all the synthetic E. coli that we simulated? Why? Because we needed to include these in an agent-based model. So in the exogaia agent-based model, as I was mentioning, it was developed by Tim Lenton and his colleagues. In this particular ABM, they were looking at the temperature and the biomass of a planet based on various chemistries and planetary albedos. And we needed the synthetic chemistries so that we can plug them into this agent-based model. And the ABM, in this case, takes as input the energy of a star, the fraction of the planet's thermal energy, the fraction of incoming radiation, that's the albedo that is being reflected, the geochemistry network, which is abstract, and I'll show you in a minute, and the connectivity between any two chemicals. And we included in this agent-based model the parameters we extracted from the synthetic E. coli networks, such as the probability of geochemistry, the number of nodes in the chemistry, the number of nutrients, the probability of gene mutations, and the probability of biochemistry. The probability of biochemistry, this is the soft spot. It's where we basically approximated the network density of the chemistries with the probabilities of the network densities of the synthetic genomes with the probability of the biochemistries.
What were the agents?
At the chemicals, the actual chemical molecules. So here, basically, this is the exogaia model, right? So so again, this was not developed by us. They were kind enough to lend it to us so that we can adapt it. Basically, you have different chemistries where you can connect them in any fashion you want. You do it in the computer. So you have highly connected geochemistries versus an example of a biochemistry. So you have geochemistries and biochemistries, biotic and abiotic. And basically, you are looking at, in the agent-based model, you generate a planet with a particular geochemical network. You can generate them all. You can see the planet with life in 100 separate times or 100 time steps, one after another. And then you test for the habitability of the planet. And the way it works more as a regulation mechanism where you have a relationship between the temperature of the planet and the micropopulation. Right? So, and you have the increase in temperature leads to increase in population, that leads to decrease in temperature, that leads to decrease in population. So it's a feedback loop. And like this in the model, they showed that you can have basically 4 types of planets. You can have a dead planet where they are clearly inhospitable to life. You can have a critical planet, planets that can be successfully colonized by life, but that life is prone to go extinct at random times, the Gaia bottlenecks that I was showing before. Then the bottlenecks, the planets that once seeded either die very quickly or continue to survive until the end of the experiment, which is again the Gaia bottlenecks. Or immortal, the planets that are always successfully colonized and survive until the end of the experiment, which are the Gaia regulations. So basically, in the agent-based model, you can look at the critical planet, how it's being, what is the relationship between the temperature and the biomass, total population of the biomass, based on different chemistries. So that would be the profile of a critical planet. This would be the profile of a bottleneck planet. Sorry, that would be, yeah. This would be another profile for a bottleneck planet. And this would be the profile of an immortal planet, right? So the total population. So basically, if we do all these simulations in many times, we can look at the long-term habitability increases as we increase the connectivity in the geochemistry and biochemistry. So that leads me back to the idea of connecting the metabolic networks for the genome, for the synthetic genomes, right? So more geological connections in the geochemistry means that there are statistically more pre-established recycling loops for the biota to exploit than for the low connectivity, right? So we have the degree of connectivity versus how many planets you can expect to find based on the degree of connectivity, how many of them are critical, how many of them are actually dead, how many of them are the bottleneck planets, and how many of them are actually immortal. So this is that model. We adapted it. So what we did is from the universal genome where we created the connectivity of the networks, we bridged this universal E. coli with the exovial agent-based model. So we informed all the parameters with the parameters that we got from the KEGG database and looking at the metabolic boxes model with, for E. coli. And the results from our model that we adapted is basically that these are the 11 synthetic plus real E. coli metabolisms. And we looked at the biomass of the planet and at the temperature of the planet. And there are 5 types of chemistries, geochemistries, that are originally from the model. And we looked how basically which metabolic networks are leading to that sweet spot between the planet biomass and the planet temperature. And basically we can see here that particularly for geochemistries B, C, and A, we can produce biomass for E. coli universal and E. coli superorganism. Superorganism E. coli is the E. coli that is feeding on combinations of 3 of the 6 gases, right? So we have the universal, which is feeding on all 6 gases, and the superorganism that is feeding on combinations of 3 of each. But if we are looking at the temperature, which basically what we can see is basically that these planets are super hot, right? So again, we have this logarithmic scale, so we have very, very hot temperatures. So basically here, if we are looking at the actual E. coli, the real one, it would not survive, right? So it would not render any kind of biomass given the geochemistries that are in the model. So these are basically, so why did we do all this? So basically we can, in this fashion, we try to look at how we can bridge microprocesses and macroprocesses to understand the profile of exoplanets and whether we can infer life. Now, life is not formed only of E. coli, right? So there are more types of organisms, of genomes that we can use and simulate, right? So this is more of a proof of concept. But we can, and basically what we did is to look at real chemical networks that we know that they are bounded by chemical rules and laws, generate metabolic networks, generate genomes, and link those with planet temperatures and biomass, and that with planetary biospheres, right? So, in conclusion for this project, basically we have only now got to a point where biological models and geochemical models are complex enough to interact at a planetary scale. Although computationally intensive, and again, thank you Google, this is the right time to couple these models and explore the large parameter space potentially enabled by machine learning. The code is available online. It's on GitHub. It's called PyAtmos. And the paper was presented at NIPS conference last year. And some general takeaways and conclusions. I think looking at the problem also from the exploratory and discovery side, or this point of view, it's really useful. You know, These are, we are playing with synthetic organisms that we don't know if they exist, and with planetary atmospheres that again we don't know if they exist. But if in the process we create a roadmap or a protocol that would help us once we do see all those atmospheres, and we can say this is the probability of finding this type of biological processes here, I think this makes a lot of headway that we can get ahead to the next instrument discoveries. Also I think that today we are the best equipped that we have ever been with methodological and theoretical tools to answer some of these big questions. Okay, so it's one thing to ask a question about how can I improve this bottle of water versus a question that is there life on exoplanets, or is there life elsewhere in the universe? And these are indeed some areas where potentially deep learning, machine learning, ABMs and other types of methods computational methods can really help, particularly when it comes to generating synthetic data, to applying new algorithms to existing data in order to find new discoveries. And actually this is what some astronomers are doing now with the wealth of the radio frequency data that they have. They have so much frequency data, but they didn't have these tools before, so now they are looking back at the data that they gathered for the past 30 years. And I think it can also enable us to test some hypotheses and scenarios much faster. And I welcome your questions and comments, and I'd also like in the end to thank not only the researchers but also the other mentors. Chris Campis is at the Santa Fe Institute, Claire McLean, she's in Oxford, Daniel Langerhausen, he's in Switzerland, Massimo Mascaro from Google, and Natalie Cabral, the head of the SETI Institute. Yes.
So, what's the Lego principle?
Building blocks. The building blocks of life. Whether if you are combining different nucleotides, what would that render? If it's not DNA based, what would that render?
I noticed one of your ABMs was called Exogaia. Do you want to talk a little bit about the origin of that name? Are you working toward modeling interacting networks of biomes with possibly some overall regulatory mechanisms on a planetary scale?
So the Exogaia model was not developed by us, was developed by Tim Lenton and his collaborators. So in that particular model, they were specifically looking at planetary biomass and planetary temperatures. Okay, so in the model, you can generate a planet with a certain geochemical network, which again is synthetic, is abstract. Chemistry is combination, is combinatorics at the end of the day. And then you can seed the planet with life at different time steps, and then you try to see whether the biomass that is being generated is being stabilized or not. It's an abstract model, meaning that you have a set of parameters, and I think there are more than 20 parameters in the model, from the brightness of the star to the albedo of the planet, all the way to how many microbes you can have as the seeding of the life on the planet. So you can tweak on those parameters with some real values, which we did, and see what comes out of that. Yeah.
So just picking up on something you just said, the protein networks alone that you're generating are factorial.
Oh yeah,
They're they're factorial in terms of the sample space.
Right.
So that alone, never mind all the other things, because there are lots of things here where so the model has 20 parameters. The space that you're trying to cover is absolutely gigantic.
It is.
Gigantic.
Yes. Yeah.
So how does a project like this tell us really anything about what's possible on any of those planets?
So again, because
First of all, what are your sample? How many planets do you generate altogether out of this simulation?
So from the Atmos code, we generated originally 150,000 types of atmospheres, and now there are more than 250,000 because the guys kept on working after the end of the project. And again, Google helped us, so we were able to generate more than 250,000. We don't know that many planets in the universe, right? As I was mentioning, now we have maybe more than 3,000, and probably not all of them have atmospheres. Going back to your question, is once you find a combination of gases on an atmosphere, as I was giving the example for that planet that was discovered a couple of weeks ago where we know there is water, what can we infer back with respect to the type of chemical processes that can happen at the surface of the planet, right? So if we are looking at the overall idea about how we can link microprocesses with macroprocesses, right? We are generating these climate profiles in the abstract. But this is not abstract. This is real, right? So the KEG database, it shows you the constraints of chemistry that are available everywhere in the universe. The exoplanetary, yeah, it generates in the abstract planetary biomasses and temperatures, but the parameters are real. The parameters with respect to the brightness of the star with the brightness of the atmosphere and the way these are interacting with each other, these are real constraints, real physical constraints. So part of it is real data, part of it is what we can infer from this real data.
So the DNA in life has base pairs, 4 base pairs in various combinations, and they code 2 amino acids, and that's how we create proteins. And I believe I've read that all life on Earth has the same code. You get the same amino acid out of the same base pair.
Yep.
But that's a path-dependent thing.
It is, and it's the universal ancestor for us is called LUCA. I don't remember what the acronym is where it's coming from. That's right.
Universal ancestor.
That's right, but this is not DNA dependent.
Sure.
This is chemistry dependent, so that's a little bit different.
But it would argue for the fact that life on the Earth started with one organism.
LUCA, yeah, that's what they call it.
And there was no, so it really is a sample of one. There was no competing strain. There's no, Asian strain as opposed to American
that's not
strain.
necessarily true. So from astrovirology, which is a branch of astrobiology, they are looking at how different molecules, basically how RNA or different molecules are competing for the transmission of information, right? So they are looking at, okay, can we transmit, why is RNA, why is this way through which we are transmitting information in a replicative sense? And apparently it is the most efficient way in which we can think, but given the conditions of Earth, right? Given the conditions of another planet, you might have you might have a different way of transmitting information at the molecular level. Yeah.
That's a theory. You gotta find
That's
several other
sure. Sure. I mean, the next missions that are going to go to Titan and Europa, which is here in the solar system, are going to be very interesting to see whether there are microbes on the moons. So
Has the Earth ever been that cold as Titan and Europa are?
I don't think it has been really hot, but not as cold as Titan and Europa. So, speaking about geochemistries, going back to the geochemistries, why they are looking at Titan, Europa, and Enceladus? Because the core of the planet is warm enough to have. So it's kind of like in the vents in the deep ocean on Earth, where you don't have light, but you have enough heat that is coming from the core and from the movement of the planet that can sustain life. So that's why they are looking at those moons in particular.
It might be warm enough below the surface.
Right. Yeah.
Or something to grow that lives up to that heat, like something lives up in
Right.
the
Right.
results.
Yeah. So, yeah.
So, do we have, one of the things that helps sustain life, protect life on Earth is the magnetosphere.
Mm-hmm.
Do we have any information on exoplanet magnetospheres?
They can infer that from the relationship of the planet with the star, but it's not by no means very, so that's actually a very good point. So we don't really know about the magnetospheres of the exoplanets. Yeah. Yeah.
A
That's
future
true.
version of the model as we get more data.
That's true, that's true. So they were saying, at some point they did find an exoplanet in our closest system, which is Alpha Centauri, which everybody is excited about, but apparently the planet is so close to the star that it cannot have a magnetic field. But, yeah, so
So when the ecoli is more universal, does it require more energy? Is it less efficient at extracting energy?
I don't know. Question, we didn't look at that. We did look at the metabolic flux. So in the metabolic flux, which is basically the biomass that you produce, it's more enhanced than the real E. coli. So by the way, real E. coli on Earth doesn't feed on any of these 6 gases, right? So it's not the way the actual real genome functions. So we modified it just to see the relationship with the specific atmospheres. But no, I don't know.
What does it mean for it to be enhanced, the flux?
So it has more output than the E. coli flux. Yeah, than the real E. coli flux. So the metabolic flux is showing you how much biomass you can produce, more or less, the microbial level.
Thank you very much.
We will be around for all the questions if we have them. Phil, just want to raise his hand.
Yeah.
Right.